cells. What do they all have in common? Well, for a start, they're all surrounded by a phospholipid membrane that holds everything together and creates a semi-permeable barrier that separates the inside of the cell from the outside. One experiment we may all be familiar with is looking at how different vegetables absorb water or leak pigment due to osmosis or changes in membrane permeability. These are classic examples of how membrane permeability functions. But do any of these experiments have any relevance in the cutting-edge world of modern scientific research? The short answer is yes, particularly in the world of microbiology. That's because one of the fundamental mechanisms of antibiotic resistance is caused by changes in membrane permeability to antibiotics, with resistant bacteria either completely preventing the entry of antibiotics into their cells or by efficiently pumping the antibiotics out once they've got in. But bacteria are much smaller than, say, a potato. So how can we test for changes in bacterial membrane permeability? Here I have two bacterial cell cultures, one with a mutation that affects membrane permeability and one without. To each of these cultures, I'm going to add a fluorescent chemical which should be excluded from the cytoplasm of bacteria with an intact cellular membrane. Once we've added our reagents, it's time to go and qualitatively analyze our bacteria under a special microscope. And so here we have samples of our two bacteria, our membrane mutant and our control. So we can see that the membrane mutant has a much higher degree of fluorescence than the control. And that's because the mutant has increased membrane permeability, meaning that more of the fluorescent dye can enter the cell. By understanding how resistant bacteria prevent antibiotics from accumulating inside their cells, this could lead to the development of chemical modulators which increase membrane permeability and could be used in conjunction with antibiotics to increase their effectiveness.